All right, Assalamu alaikum. So let's get started. This is the second. This is part two of the chapter 8 slides I'm gonna upload, and here we're gonna talk about biological membranes. Right? We've just started talking about this last time, and we inshallah hope to finish it today. Yeah. So biological membranes. Okay. So as we talked about last time, biological membranes are described by this fluid mosaic model, where the membranes are very fluid in nature, like almost like oil, and then you have this mosaic-like things sticking out on the membranes and so on and so forth. And we talked about the various components. You have the integral proteins, you have the peripheral proteins, you have the oligosaccharides or glycolipids. You have proteins that are anchored by lipids, so lipid anchored proteins, as well as you have the proteins that have uh, glycosyl moieties, or we call them glycoproteins, right? So we saw them last time. Same thing here, shown here. Um, Again, this would be a peripheral protein. This would be the peripheral protein. This would be the integral proteins here, shown here, right? And so on and so forth. And then you find that some proteins are anchored to the membranes. These are anchored. We call them anchored, and we'll talk about them today. Anchored proteins. Um, okay. And of course you have this glycolipids, you have glycoproteins, and so on and so forth. But also what you have, interestingly here shown nicely, are these guys, these these guys, right? So what are these guys? And if you look carefully, these look like lipids, of course, and in the next slide is actually shown properly. These actually are cholesterol. So it turns out cholesterol is an integral part of biological membranes, right? It's very important. It serves a very important function. And if you recall, cholesterol is also, so these guys are amphipathic, right? Amphipathic. They have a polar head and a non-polar tail. Polar head, non-polar tail, non-polar tail, right? Same thing, cholesterol also has a polar head, and they have a pretty much non-polar tail as well, and that's why they can actually fit in very nicely in biological membranes. So this would be the polar head of cholesterol, and this is the phospholipid polar head, polar head, polar head. Okay. Um, so what does cholesterol do when they're present in in biological membranes? So one of the things that you should really know and be very comfortable talking about is that cholesterol affects the fluidity of membranes. It has a direct effect on the fluidity. And if you recall what fluidity means, it means the how liquidy the membrane is, right? Um, so it has, and what's interesting is cholesterol has this effect. It has a different effect at different temperatures, which is really interesting, right? So, for example, if you have, you know, a different effect at different temperatures. So it turns out at warm temperatures, at warm temperatures such as 37 degrees or higher, cholesterol prevents movement. It restrains the movement. It prevents the phospholipids to move too much, and so it prevents fluidity. It decreases fluidity. So let's see. It decreases fluidity. So it decreases fluidity. So at warm temperatures, cholesterol decreases fluidity. At cool temperature, it maintains fluidity. It increases fluidity. So that's really interesting. So please make sure you understand that at cold temperature, cholesterol increases fluidity. At warm temperatures, cholesterol decreases fluidity. Right? And this is shown here um, in this slide that. So let's see if we can do this at low temperature. Let's see if we can circle this. At low temperature, cholesterol in so of course what happens is at low temperature, if you don't normally the membrane would be less fluid. It'll freeze, like oil freezes at low temperature, so the fluidity will be decreased. But if you have cholesterol present, it increases fluidity. At high temperature, of course, as you would imagine, if you have no cholesterol, the oilish, like biological membrane, the fluidity would be increased. It'll be more liquidy. But if you have cholesterol, cholesterol actually causes the fluidity to decrease, right? So please make sure you guys watch this video here. It's a link. It's a very important link. I will test you about this in the exam as well. So please make sure you understand this concept of how cholesterol affects membrane fluidity. And in fact, that's shown here as well. So remember we talk about this melting temperature or phase transition. So if you have, so that's shown here. So if you have normal biological membrane, if you increase the temperature, there's a sharp transition point where it becomes, this is the crystalline phase. So your lipid becomes, so here for example, so let me write properly. So at low temperature you have solid or crystalline, and this is liquid or more fluid state at high temperature. So this is what you normally see, that gel to liquid transition. So solid, I mean gel, right? And this is liquid. So as you increase the temperature, it causes disruption of this phospholipid interaction, hydrophobic interaction. It causes a sharp transition from gel state or solid state to a liquid state. Now if you have cholesterol, and that's shown here, what cholesterol does, it prevents this sharp transition. It actually serves as a buffer, right? And that's what they're mentioning here, that the blue light here, so the blue curve shows the fluidity in the presence of cholesterol, and really it acts like a buffer. So acts like a buffer, and prevents the sudden transition from a sharp to a gradual, right? So this is very important, of course, because if the temperature were to increase for an organism, you don't want biological membranes for the cells to fall apart very suddenly. So there is a protective effect of cholesterol, right? And that's what's shown here in this blue line here, in the presence of cholesterol with cholesterol. Okay. All right. So moving on again. These are the cholesterol things that are integrated into membranes. So you have. So please make sure you understand all of the components. If I were to ask you to draw a mosaic fluid mosaic model of biological membranes, make sure you show this double lipid bilayer. Make sure you show integral proteins, are these or these. Make sure you show peripheral proteins. Make sure you show glycolipids. Make sure you show glycoproteins and cholesterol. So all those things have to be mentioned. Okay, so moving on again, this is the same thing. This is explaining again the role of cholesterols here, of course, integral proteins, carbohydrate groups that are on it, glycolipids that are there, peripheral proteins as well, right? And I think what they're also showing you that there are, what these are the peripheral proteins, but they're also showing these things, the presence of these sort of structures that are shown here, and these things. So these are membranes actually are linked with what's called cytoskeletal parts, right? So it's called cytoskeletal. So the membranes, skeletal, structures. So there are these filaments and tubules that things are connected with, and that is actually seen here better, the cytoskeleton part. So that's just for your understanding. Of course, it's important, but you don't have to draw it. If I were to ask you to draw it, if you draw it, that's great, but you don't have to. What you have to draw would be this part here. But if you draw the cytoskeleton, that'll be great as well. But you don't necessarily have to. But make sure that you have microtubules and you have these filaments and stuff, which are really important and integral. And which is also again shown here. This is the cytoskeleton pinning, again, things like actin. Actin is a protein. It forms filaments. We call them actin filaments. These are important to maintain the integrity. But then, of course, you have these, peripheral proteins. Peripheral proteins. You have integral proteins. You have phospholipids, of course, all over the place in here. You have cholesterol sitting in here. You have glycolipids shown here. And so on and so forth. But what's really interesting also is to understand that if you have a cell, and this is a cell membrane. This is a cell membrane, right? There are proteins which are receptors, right? There are also proteins which are channels, and we saw them in the previous slide, right? But there are also proteins that are involved in cell-cell junctions. So if you have a cell, another cell interacting with this cell, so there are proteins on both ends. They interact with each other. They could be like receptors to bind onto other cells. They could be involved in what's called extracellular matrix. If you have, for example, if you take a look at a liver as an organ. So liver as an organ is made up of many cells, right? So many cells. So how are the cells, held together? Because if you take a liver, it just doesn't fall apart. They are attached to other things, and these are the interactions that are involved in holding on to other cells and creating what's called extracellular matrix and cell-cell junctions. So these are important concepts for you to understand and keep in mind as you study other things in the future. Okay. So when talk about membrane proteins, again, we said there are different kinds of membrane proteins. There are peripheral proteins, there are integral proteins, right? And so this would be an example of an integral protein, right? And then these would be examples of peripheral proteins. And peripheral proteins are bound by electrostatic interactions. They can be removed by raising the ionic strength of buffers. So for example, if you want to purify this protein, for example, you put a high salt buffer, and this high salt buffer would disrupt these ionic electrostatic interactions, and then you can actually purify them. But these integral proteins, these are really very tightly bound in the membrane, right? They're tightly bound in the interior of the membrane. And these are hydrophobic parts. They're interacting with lipids, hydrophobic parts. So they can only be removed by treatment with detergents, right? So detergents, we didn't talk much about detergents. Actually, we did, right? These are soaps. Detergents are soaps. And we talked about how soaps are. Soaps are this fatty acid that have a negative charge, and they are sodium salt of a fatty acid, right? So what detergents do, they actually can form micelles with these proteins inside micelles, right? And that's how the detergents can be used to purify integral membrane protein and keep them in a proper environment, right? So soaps or detergents. So please make sure you understand that as well. The book talks about it, so please, I'm expecting you to read the book as well for this. Okay, now I have talked excuse me, I have talked about how proteins can be anchored to membranes, right? Um, and there are basically many ways that a protein, like this is a protein, this can be stuck to the membrane, right? And there are basically we'll talk about three different anchors. So three different anchors, and the word, of course, anchors, right? The whole idea that you attach something to them. So they can be anchored, number one, by what's called N-myrcylation. So N-myrcylation, and here the N-terminal glycine. So the first amino acid has to be a glycine. So this is a glycine. So if you look at this is the amino group, this is the peptide bond, right? And this is the R group. You have two hydrogens, one up, one down. So this is glycine. So what happens? Glycine, the N-terminal glycine, the amino group forms a amide bond with carboxylic acid, right? And this is a carboxylic acid, right? And this is a it forms an amide bond, and this carboxylic acid is normally done with a C14 amino acid, which is called myristic acid. So if you go back to the fatty acid slides, you'll find a C14 amino acid called myristic. So what happens really is this is the protein. You have um, the amino group is glycine here, right? So this will come in and react with a C14, so CH2, which is so you have 12 times and you have CH3. So C14 amino acid is going to C14 fatty acid is going to react with it, and what you would get, you'd get this protein that has CH2 and NH, and then you lose the water. Water is eliminated. It's the same sort of reaction that you see all the time, and then you have CH2 12 times and you have CH3, right? And this is the example of a protein which is linked to a lipid, and then this lipid, oops. So this lipid, oops, I'm sorry about this again. Okay. So this lipid, I don't know what I'm doing. Okay, so this lipid then goes to the membrane, right? That's how you anchor it. So that's called the first one is called N-myristylation. It uses myristic acid. That's an important point, and it needs N-terminal glycine. Okay, the second one is links an internal cysteine with palmitate, right? So you have a protein that has an internal cysteine. So cysteine is BCH2 SH side group, right? And this reacts with palmitate. So palmitate would be, so let me just make it nicely. So this is SH. A palmitate is a C16 acid. This is palmitate. So C16 would be CH3, CH2 many times, and in fact 14 times, and then you have COOH here. So you have this water being eliminated, and what you form would be CH3, CH2, 14, C double bond O, and then you link it with S and CH2, right, of a protein. So this is called a thioester bond. Thioester bond. This is a thioester bond, and this is the internal cysteine as shown here, thioester bond. Okay, so first one, oops, sorry about that. So first one is N-myristylation. The second one we talked about is thioester with sulfur group here. Okay. So this if you look at sulfur here, and this would be example of S-myristylation, right? It's normally with cysteine, but sometimes it can be with serine as well, but normally cysteine. Or you can have an N-myristylation with an amino terminal glycine, so you have glycine forming an amide bond, right? This is cysteine. Okay, so please make sure you guys know this. And then there's another one, farnesyl, and we'll not talk about this farnesyl, so don't worry about this. The third one, so we've talked about number one, number two. The third one we'll talk about is this scary guy here. It does look scary, but we'll talk about this, and you'll see it is not that difficult to understand. So this was what's called a GPI anchor. Okay, so I'll save this into a new slide, and I'll stop here for now. Thanks.